Innate immunity, physiological barriers. So, anatomical barriers that comprise skin and mucous membrane try to prevent the microbes or foreign antigens to enter the body. But if they do enter, then the next barrier that they have to face is that of physiological barriers. So, let us look at the learning outcomes of this session. Conditions like temperature and pH contribute to innate immunity. Soluble as well as cell associated molecules to contribute to the same. So, in the innate immune system, molecules play an important role. Molecules such as lysozymes, interferons, complement components are able to non specifically eliminate antigens that have been able to get through the physical barriers like skin and mucous membranes. The innate immune system has certain molecules that are able to recognize foreign particles by pattern recognition. And the physiological barriers of the innate system enable almost immediate eliminations of the antigens. So the response may not be a very highly specific response, but the response is non-specific and fast. So let us look at the general conditions in innate immunity. So the body temperature is something, or the temperature itself is something that is important in innate immunity. Like for example, chickens, they are immune to anthrax as their body temperature is high. So because the body temperature is high, the causative organism is not able to grow in the chicken. It has also been observed that environmental heat stress or cold can influence innate immunity. So it is not just within the body the temperature that matters, but even outside temperature does affect innate immunity. For example, in very cold conditions, it is possible that the immune system is suppressed. But under other conditions, at a cold condition where the animal or organism has already adapted to the environment, there the cold condition may not affect the innate immunity. Now, rhinovirus replication is limited at warm temperature. So, when you have in the mouse airway cells the temperature being warm, then it has been found that rhinovirus cannot multiply. So, therefore, this is a very direct link of the fact that if the temperature is high, then the causative organism may not survive that high temperature. Cold-blooded animals have been found to move to warmer climates when they are ill. Another condition that can affect the growth of the microbes is pH. For example, the gastric pH, which is around 2 to 5, is not at all conducible for the growth of microbes. So suppose you have microbes coming in through food and it enters the stomach, then within the stomach there are all chances of the bacteria or the pathogens getting killed because of the pH, very low pH. Extracellular acidosis, that means high proton content, uh, extracellular, influences the innate immune components. So the presence of a certain amount of H plus in the surroundings of a cell does affect the innate immune components and thereby the response itself. And now we look at the soluble factors of the innate immune system. So for example, let us consider lysozymes. So lysozymes are enzymes that are produced by tears, saliva, mucus, epithelial cells. These all form what is called as lysozyme. And what is the mechanism of action of lysozyme? The lysozyme is able to break the glycosidic linkage between N-acetylglucosamine and N-acetylmuramic acid, and it is able to uh, um, cleave the beta-1,4 glycosidic linkage that is present. Now here what can be very clearly observed is that when you have the lysozyme cleaving or breaking the cell wall, it also leads to certain peptidoglycan molecules to be released in a certain combination. So you have here the amino acids linked to NAG/NAM. So this in itself is a very specific pattern that is released. So lysozymes, apart from actually degrading the cell wall and killing the bacteria, can also release broken parts of the peptidoglycan, and that itself is recognized by several other immune cells. And that leads to enhanced phagocytosis and inflammation. So you can see how lysozymes have a dual role, directly acting as an antimicrobial and indirectly, uh, basically enhancing the immune system. Bacteria may modify their peptidoglycan to resist lysozyme. This is a phenomenon that has been observed in certain bacteria. Now, the second example is that of interferons. We all know that interferons are antiviral molecules. And what has been found is that interferons are actually synthesized or in response to a virus that has infected a cell, you have interferons being released by that particular cell. And the interferons that are generally present by virus infected cells are the type 1 interferons. So there are two basic types of interferons, type 1 interferon and type 2 interferon. Another type 1 interferon comes interferon epsilon, interferon beta, etc., interferon alpha, etc. While type 2 interferons contain gamma interferon. Now, this interferon that has been secreted from that cell which contains the virus, this interferons can go and bind to interferon receptors on the neighboring cells, and in response to the interaction of the interferon with the interferon receptor, there is a signal transduction, and based on the signal transduction, you have incapacitated the multiplication of the viral genome. That is because the neighboring cells which have been activated by interferon are able to produce antiviral proteins, and because of the presence of these antiviral proteins, you have the virus replication halted. And with no replication of virus, you cannot have packaging, and then for the population of viruses will decrease. But what is interesting to note is that gamma interferon or even alpha beta interferon are also able to uh, enhance somehow the activity of the cell mediated immunity. So this that was observed is innate immunity, and this because it's cell based is what is called as adaptive. This is short term protection, this is long term protection. Okay, going back to the fact that if you have these gamma interferons present, it can activate T cells, it can also activate B cells. But most importantly, it can activate antigen presentation by antigen presenting cells. So when they present the antigen contains the um, interferon gamma. So that interaction of interferon gamma and the surface interferon receptor will lead to the dendritic cell or macrophage getting active, and on activation they give rise to MHC class 2, and on the surface they present the broken viral components exhibited on the surface, and then one knows that you can have adaptive immune system happening. So you can already see that how T cells and B cells directly are getting activated by gamma interferon to synthesize or produce more of their videos. Type 1 interferons are produced by virus infected cells. Type 2 interferons are secreted by T cells, natural killer cells, and macrophages, and enhance antigen presentation to activate adaptive immunity. Type 1 interferons bind to surfaces of neighboring cells, inducing a signal which then leads to production of antiviral proteins. So this is just reiterating the fact that in innate immunity, you have short-term protection because of the production of the antiviral proteins in response to the interferon produced by the virus-infected cells. Another example is that of complement system. Complement system is basically a set of proteins that are present in the serum, and these set of proteins are present in an inactive state. but it can be induced by foreign particles to become active, and in the activated form, it eliminates the microbes by affecting the outer envelope, basically the membrane of the pathogen or the microbe. So what the complement system is able to do is lyse the pathogens by damaging its membranes. It can induce inflammation. So by inflammation, we all know that there can be elimination of microbes. You have opsonization. So this is very clear that binding of the complement to the foreign particle is actually calling in several other molecules of the immune system and cells of the immune system to basically eliminate the antigen that it has bound. You also have what is called as the immune clearance. So basically, in all, the complement system is eventually going to try to decrease or try to eliminate the foreign particles that have entered. Now, the complement system is observed to be a cascade of events that finally leads to damaging the membrane of the pathogen. So, as you can see over here, the complement system has been able to form a pore in the cell membrane, and therefore, you can have extracellular components entering the intracellular space, while intracellular space can go out. And so, you have an osmotic imbalance created because of which the microbe or the pathogen can die. Now, what is very important in innate immune system is what is called as pattern recognition receptors. Let us look at what we understand by pattern recognition receptors. So innate immune system has certain recognition strategies, and this strategy involves ability to recognize patterns that are present on the microbes. Now, what kind of patterns? So the patterns can be formed by various different molecules present on the surface of the microbe. So those patterns that are formed by molecules present on the surface of the microbes, they are called as pathogen associated molecular patterns. Now, interestingly, if you also have cells that are damaged and they show and those damaged molecules or sorry damaged cells which need to be eliminated from the body also show on its surface what is called as damage associated molecular patterns. So the innate immune system is able to recognize both PAMPs and the DAMPs. Sorry, over here it should be DAMPs. And this is important because these molecular patterns being recognized by the innate immune system, it is able to generate an response very fast. So these patterns that are present on the microbes or on the damaged cells are very different from that present on the normal cells, and therefore you're able to easily distinguish between the two. Now, this pattern or these patterns that are present on these cells can have to be recognized by something, and those molecules are pattern recognition receptors, which can be soluble factors or they can be bound to the surface of the immune cells. So, they can be associated with the cells or they can be freely moving. Now, pattern recognition receptors can associate or interact with PMPs, and they can also interact with the DAMPs, so the damage associated molecular patterns. So the PAMP PRR, that is pattern recognition receptor, and the pathogen associated molecular patterns, they interact uh, their interaction leads to signal transduction, which in turn induces synthesis of cytokines and chemokines. And we all know that cytokines and chemokines can activate a range of immune cells and molecules. Then you also have the toll-like, you also have the best example for pattern recognition receptors is the toll-like receptor. Now, this toll-like receptor, when it binds to certain molecules present on the microbe, that is, it could be a particular lipopeptide, or it could be the lipopolysaccharide in the gram-negative bacteria, or it could be flagellin of a certain bacteria. So, these constituents of the bacteria are recognized by the toll-like receptors, and being transmembrane, they are able to successfully transduce signal through a certain pathway and eventually leading to expression of certain genes in response to the binding of the toll-like receptor to the patterned molecules that are present in the microbe. Now, this is the toll-like receptor. So, as you can see, it has a transmembrane domain, it has an ectodomain, and in the ectodomain, it is able to bind to the lipopolysaccharide of the bacteria. And the ectodomain is also having a leucine-rich region, which recognizes the pathogen associated molecular pattern. The intracellular domain initiates the signal transduction. So when you have the LPS coming and binding to the toll-like receptor, then what is immediately observed is that the intracellular domain is also going to initiate a signal in response to what the toll-like receptor has bound. Other than toll-like receptors are present node-like receptors. So you can see how many different family of the node-like receptor is present. So they all can start, um, you know, acting as those molecules that are able to recognize foreign particles. Similarly, you have RIG1 receptors, like receptors. So when the viral mRNA comes in, then the RIG1 gets kind of active, and over a cascade of events, what you'll observe is that you have certain regulatory factors that are able to induce the production of uh, alpha interferon to influence the innate immune system. So let us make the conclusions. The temperature levels of the body and the pH of various fluids or secretions can lead to inability of the pathogens to grow or survive. Lysozymes, other than being antimicrobial, can also mediate the innate and adaptive immunity in response to a pathogen. Soluble factors like interferons, complement proteins, collectins, etc., can rapidly eliminate pathogens by different molecular mechanisms. There are several molecules called pattern recognition receptors, either present as soluble factors or associated with cells that can recognize molecules through specific molecular structures on the pathogens. Therefore, the physiological barrier comprises molecules that are able to establish contact with pathogens due to specific molecular structures on the surface or within the microbes. Many microbes have found ways, of course, of evading this recognition. Thank you.